thanks for the opportunity for us to make this presentation this afternoon. We're going to talk about a framework for determining the physiological benefits of dietary fiber. And I think the keys to understanding or keys to determining this is determining these physiological benefits is going to be very important. Why? Well, without any benefits, I don't think we'd be talking about dietary fiber labeling at all. So to get it in the first place, fiber had demonstrated benefits. Second, what do we want to do? We want to define adequate physiological effect, quality and quantity, not in strict terms, but in what would constitute, say, solid science or procedures for doing this. Our immediate concern, of course, is the Codex definitions 2 and 3, which have to do with either isolated or synthetic fiber sources, and also, of course, the proposed U.S. regulations, which parallel quite closely to the Codex definition. And then how do we going to do this? Well, of course, what we need is a harmonious approach and tools that would be used for determining adequacy. I do want to make a note. We will be recording the panel discussion at the end because we're going to put the notes together regarding that panel discussion. And Courtney is fantastic, but even she can't write that fast, so we're going to record it. Second thing, to address this topic, we've got 3 speakers today. We've got Nicola McKeown from Tufts, who is going to be talking about the dietary fiber database that ILSI has initiated. And I think you'll find some fantastic information there. We're going to have Julie Jones from St. Catherine's University talk about the impact of fiber variety and beneficial effects. And then Stuart Craig from DuPont is going to talk about the regulatory perspectives. And then we're going to finish up with a panel and open discussion where everybody can have their say and input and presumably get their questions answered. So we'll start off with the 9 Vahouny health benefits, launch of a new database with Nicola. And here's the questions that we do want her to address. What was the objective criteria for developing that database? Public access to encourage consistency will be part of it, how it would benefit rulemaking, and what kind of consumer education will come out of this. And then finally, it'll be used as a research tool for current stakeholders. So with that, I'll turn it over to, and a tool for guiding future research. I don't always remember everything I write. Okay, so Nicola.
Thank you. Well, thank you very much for the opportunity to be here today to speak to you. This is my first Vahouny meeting. I've learned a great deal. Let me walk you through some of the key reasons why we undertook this project. ILSI put out a call, the Carbohydrate Committee, for this database development, and when I saw it, I thought it would complement my research very nicely. I listened to Dr. Slavin talk the other day, and I think she said you can either be optimistic or crazy when you undertake large meta-analysis. I haven't decided which field I fall into, but certainly I'm not in the crazy yet. So we've heard a lot over the past couple of days about fiber and the health outcomes, and how fiber is different. And Dr. Slavin nicely described how much variation there is in human studies as they pertain to the health benefits of dietary fiber. There's certainly different types of fibers that are administered. There's variation in the experimental design, and of course variation in dose and duration. And so these were key aspects to consider when we started to develop the database. And I'd just like to say at this point, in terms of the database, we're talking about a scientific database, not a nutrient database. So I don't know why my formatting is a little off, but in terms of the Codex fibers, you know how they're classified as fiber Codex 1, 2, or 3. I won't spend time going through those definitions. But what was important was to look to see, in terms of this database, what constitutes a physiological health effect. And we used the 9 Vahouny fiber health effects that were agreed upon at the 9th meeting. And they are total and LDL cholesterol, postprandial glucose and insulin, blood pressure, increased fecal bulk and lactation, transit time, colonic fermentation and short fatty acid production, modulation of the colonic microflora, weight loss, weight maintenance, and reduction in adiposity, and the last one there, increased satiety. So those were the 9 outcomes. And so the step was, if we want to link dietary fibers to health outcomes, we need to compile and synthesize the available body of literature. And so there was 2 steps in this project, and the first step is ongoing. So it's to create a database that systematically compiles and provides access to the science linking dietary fibers according to Codex fiber type designation to one or more of the 9 potential health benefits. The next step, once this database is available, would be to develop what's called an evidence map, and I'll describe that a little later. It's fairly new in nutrition epidemiology, this methodology of creating evidence maps. But what this basically does is describe the quantity, the design, and the characteristics of the research in the field, and in this case, the field of dietary fiber. Some of the objectives of having a publicly available fiber scientific database is that it would help facilitate a better understanding of how different fibers are characterized in studies. It would facilitate identifying gaps in the current research. It would create a database that would serve as a foundation for conducting future evidence-based reviews and meta-analysis. So what this database really is the first step before you would go on to create a evidence-based review or meta-analysis. It would efficiently assist researchers in identifying fibers of interest. And over the long term, and this is particularly appealing, is that we're not going to keep recreating the database. We would add to it. It would be an annual updated evidence of fiber literature. To do this, we chose to use the platform for systematic review data repository. This is free, publicly available. It's a web-based website, and it was developed by the Evidence-Based Center at Brown University. It really allows for ongoing critique, updating, and revisions. It's very flexible for creating extraction tools, so for you to extract information from scientific papers. And for the purpose of the fiber database, we use the PICO paradigm, which is collecting information on the population, the intervention, comparator, and the outcome for all papers relating fiber to those 9 outcomes. So just to walk you through the steps of what's required to create a database. The first step is to create a search criteria. And the inclusion at this stage was English language, an abstract or keyword had to contain a fiber term and one of the 9 Vahouny health outcomes. We included designs from observational, intervention, and a limited number of animal studies. We decided to include animal studies because they're the vast majority of the evidence linking colonic fermentation and short-chain fatty acid production or modulation of colonic microfloras from animal studies. And so I won't go through the list of the designs that were included in the search, but the exclusion criteria were reviews, bibliographies. If the studies were prevalence and cross-sectional designs, we excluded those. And as I pointed out, there was this selection of animal studies for 2 of the outcomes, but otherwise we did not include in this first phase of data extraction. We conducted our search in Ovid and selected published papers in Medline, CAB Abstracts, and Cochrane Central Registry of Controlled Trials. And once we had the list of abstracts available, we went through the process of screening. Any of you have had the opportunity to do this. This is a very laborious stage of the database development, but it's made easy by the fact that, or easier by the fact that we have a platform, again, developed by the EBP Center at Brown University, which allows you to pull in the paper in an abstract form. We had data entry students who are at this point helping with the screening. And just to give you an idea, at the bottom here, they could select it or they could reject it. And we had a lot where there was questions. They weren't sure based on the abstract if it should be included. Another nice feature of this particular database or this ABSTRACKR is that you can tag studies. So we use this opportunity to tag any of the animal studies that we did come across, and in vitro and observational. So we collected that information. Once we selected the abstracts, we went on to do a full review of the included abstracts, and let me walk through the review process. We had about 12,000, just under 12, that we had pulled into OVID. There was some exclusion at that stage to reduce the number of abstracts that were screened to about 7,000. Of this, we excluded about 5,000 based on the screening and included 2,000. And you can see we're down to here about 770 papers that we'll be extracting data from. There is 525 pending, so this does not include, it's not added to this yet. And the reason these are pending is because we don't have access to we're waiting for the papers to come in because we had to order them through a library. So there's been a number of steps, and just to point out, we're also going to include those that may have been missed through this. As you can imagine, it's a huge undertaking from 12,000. There are going to be ones that are missed, and if they're identified in a hand search, they'll be included. Just to give you some reasons at this point, these 770, we tracked why we excluded, so that's a great feature to have. And because of the huge amount of data, we really made some quick decisions on what to include and exclude. We'll go back to reevaluate these, but like, for example, animal studies and the observational studies were excluded at this point. We excluded some studies if the intervention was not sufficiently controlled to measure the effect of fibre. That would be if you had a fibre and a fish oil, for example, that you couldn't ascertain if the effect was due to fibre. Maybe fibre dose wasn't reported. Maybe the intervention had no control arm. We had some population exclusions. And then, of course, some reviews came in and cross-sectional prevalence studies ended up in that final exclusion. So at this point, you know all what I was thinking. If you know they wanted me to use all of this information, I would never have asked for it. I said to myself, oh, this is a bigger undertaking than I first imagined. But having said that, I think it's an exciting opportunity. I think once this database is available and out there, annually updating it won't seem as overwhelming. We have to create an animal database. We have to create a database that would extract observational. So still plenty of work in this area, which means that I might be employed for a considerably long time. So in terms of prioritizing the papers to be entered into the database, we did that, and now we're in the process of building the database. So let me walk you through when the data entry folks get to pull in the papers. What you have is, this is just an example of a paper that's in the repository. The project manager is able to upload studies to SRDR using the PubMed ID, and then assign the studies to team members who can then go on to do the data extraction process. A nice feature of this is that you can see where the progress is on the paper. So some papers are really easy for the data entry to extract the information. Often we have weekly meetings where a progress might be only 50% on a paper because they need some clarification. It could be, how do I sign this Codex? Is it 1, 2, or 3? It could be to do with the study design. So we can keep track of how the progress is coming along. And then once we get to the paper, the first part of, after you pull in PubMed, it just brings in the reference for you. And then at this point, we designed this part. So this is the data extraction part, what we thought would be important to capture. And so the first thing is the design. We have over here, study design and blindness. So it's a pull-down menu. They could choose randomized control trial, randomized parallel, non-randomized control trial, or some other. And if they selected other, there's a free text, and the data entry folks would enter in what kind of study it was. Single blind, double blind, unspecified, or other in terms of blindness. And then we were interested also being able to capture this. What kind of study design is it? Is it a weight loss? Is it an isocaloric weight maintenance? Hypercaloric? For those that are acute feeding studies, we thought it was important to capture that information. And then, of course, unspecified or other. Again, when it's other, they can enter free text, which is nice for us to be able to look at that. And the last question that we were interested here was the level of feeding control. As scientists, maybe this is where I'm interested in. You want to have a sense of maybe getting an idea of the quality of the data. Was all the food provided, for example? Was it a metabolic study, or was it partially provided, or was it based on recommendations? So increase your fiber by X grams per day. When we started the database development, we thought we nailed it. We got it up. We had a practice run, few practice runs, but then realized, what if they've more than 2 exposures or 3 exposures? So we can capture up to 4 exposures, dietary fiber exposures. And often is the case, as you know, there's dose-response studies where they've given different doses of the same fiber. We have captured that information. Let me just go here. The data entry person assigns whether it's a natural food, raw food material, or synthetic, assigns the Codex. There's a pull-down menu here where you can specify fiber type, and I think there's something like 50 or 60 options for that. Here we have beta-glucans, and what we realize now is we've modified this to identify oat versus barley. But this just gives you an idea. It captures the exposure, the duration. We wanted to know how it was administered. Was it a single food, a powder? Was it a beverage? And then just some general information about the study here would be the food form. In case these ever were of interest to other scientists, and in terms of total dietary fiber in the diet, and if they provided any information on insoluble or soluble, and then the overall energy intake, whether that was for the day, the food, or the meal. So we're capturing that data too. I won't spend time on this. Just again, this is the comparator then. What do they compare the intervention to? And then for the outcomes, as you can imagine in these papers, well, we have the Vahouny 9 outcomes that are a priority, but we might also want to just, in a free text, capture what other outcomes, if there was minerals, looking at mineral bone measures. So what we've done is we have 8 categories, health outcomes, and they all look like this. I'll walk you through this first one here. So the outcome here, the data entry person can pull down, it was cholesterol. They describe the units, whether it was fasting, and then just tag it to a Vahouny outcome. We have other, and of course if it's other, then they can specify what that is. Here we have the outcome effect. This is a free text, and really for internal purposes at this point, if we provide a summary table in the publication, did this have an effect, no effect? It was just a free text that they took from the abstract to let us know what the overall conclusion of the paper was with respect to that outcome. And the last step of this is to create an evidence map, and this is somewhat of a learning curve for our group. We recognize the importance of this. For those in the audience that aren't familiar with the evidence map, what it is, it's a method of identifying, organizing, and summarizing scientific evidence on a broad topic. It systematically examines the extent and range of research activity, but it does not summarize the results. It's valuable in areas of fast-paced research where gold standard reviews and meta-analysis can become quickly outdated. It's very useful for identifying how published reviews fit into the field. So it's becoming more valuable in nutrition epidemiology to use this. It captures a mix of studies, and it really does identify research gaps and research priorities and questions. So let me show you what some pilot data. The database has currently 500 papers that have been entered. We stopped here at 174 papers about a month and a half ago. We prioritized taking the first Vahouny outcome, which was cholesterol, and selecting those papers to present as the outcome. And so what you can see here is just based on what's available in the database at this 174 papers, 66% of the studies had isocaloric or maintenance diets, 22% had weight loss, and the remaining would be other types of designs. The equal distribution almost in terms of parallel versus crossover. In terms of study characteristics, mean age was 48 years old. 58% were men. The BMI, the majority of studies have been in overweight study participants, about 15% in obese and 20% in normal weight. And you can see just in terms of the region of publication where the vast majority of the work just for these 174 papers is being conducted. North America with US leading and then Canada. And then in terms of Europe, Finland and UK are the top two there. So as I mentioned, there's a huge list of fiber types. I'm not going to present the data for the evidence map on all of these, but just the top fibers, which are listed here in red. But it just shows you all of these have looked at the relationship between these exposures and lipids. And then we had to deal with, combination fibers. What do we do there? We certainly capture the data, and this just gives you an idea of the different combinations that have been examined in relation to lipid outcome. So this is what part of the evidence map, in addition to giving you quantifying the data in terms of tables, it also provides an opportunity to give a visual display of the evidence. And this is, as I say, restricted to these limited number of fibers. But what you can see here is each dot represents a study. The smaller the size, the smaller the sample size. And this is it just displaying it in terms of raw food material, synthetic, and natural food. And in fact, here for oat bran, when I looked at these data, realized that this is a data entry issue, and so that should be a green. But again, in terms of even data cleaning, it will give us an opportunity to look at the data in this format and make some corrections. Very busy slide here, but if you wanted to look at the type of fiber and you were interested in oat beta glucan, again, just keep in mind this is for the small number of papers. You could then use this database to see where these studies have also looked at other outcomes. So, for example, here, psyllium, a lot of blood pressure measures and weight and adiposity in those papers. If you were interested in colonic fermentation at the moment, psyllium. But you can imagine as we bring in the papers and upload the data, you'll see the shifts. And that's what's nice about this. Annually, you can see progress. At the moment, you'll just see where you stand currently. But if you could imagine annually, if this is updated, you see where there's more data coming out to support, like, for example, satiety or the postprandial insulinemia. You'll also see then where could we actually implement a larger study in this, and that might be helpful too. So with that, I just like to end and acknowledge my research group and my boss there, Paul Jakes, and then some of the students who've helped with this phase of the data extraction. And I welcome any questions that you may have.